Korean. The title is In Vivo Folliculogenesis by Trichogenic cells. So please.
Thank you for kind introduction. It's a great honor to be invited to speak in this wonderful meeting. However, it is quite stressful to speak in English early in the morning, especially after heavy drinking overnight. Today, I would like to talk about in vivo hair follicle neogenesis by trichogenic dermal cells. I'll briefly introduce hair follicle neogenesis and trichogenicity, and also I'll review some in vivo models for hair follicle reconstitution, and I'll also summarize some recent reports and our unpublished trials to preserve trichogenicity of cultured dermal cells. As you know, the hair follicle neogenesis by cell implantation is a promising option for alopecia treatment. And this is based on the hair inductive property of follicular dermal cells, especially dermal papilla cells. However, trichogenicity of dermal papilla cells is lost during in vivo cultivation. Therefore, strategies to preserve trichogenicity of dermal papilla cells need to be developed for potential therapeutic applications. Also, trichogenicity of cells need to be evaluated in animal models. To evaluate trichogenicity of trichogenicity of cells, there are several models, and especially patch assay, epidermal cells and dermal cells are combined and injected into the sub-Q. For chamber assay, epidermal cells and dermal cells are combined and incubated in the silicone chamber for a month, usually. And for flap assay, grafting epidermis and grafting dermal cells are put into the host mouse under the host skin flap. And for sandwich assay, dermal cells are put into the between epidermis and dermis of afollicular skin, and the combined skins are put into the back of nude or skid mouse. So this slide shows chamber assay using neonatal dermal cells and epidermal cells. And so as you see, hairs are coming from as early as 3 weeks, and yeah. Also, flap assay, trichogenic dermal cells and epidermis are epidermis with trichogenic dermal cells induce hair follicles. However, epidermis only does not induce hair follicles. This slide shows patch assay data using epidermal cells and dermal cells. This assay requires little labor and yields rapid evidence of hair follicle formation usually from 2 weeks after cell transplantation, and it is quite cheap to perform. So you need fewer cells, and you can inject multiple sizes per mouse. And I'm going to move to the recent reports and our unpublished trials, especially focusing on supplementation of necessary factors and spheroid cultivation and forced expression of putative trichogenic genes. Recent studies showed that supplementation of necessary factors, for example, soul skin keratinocyte conditioned medium and Wnt3A and BMP6 and foreskin keratinocyte conditioned medium and bio as GSK3 beta inhibitor and Wnt10B and also vitamin D3 analog calcitriol, they are effective to obtain trichogenic dermal papilla cells. Recently, we asked whether trichogenicity of dermal cells can be increased by supplementation of outer-sheath keratinocyte conditioned medium. We found that by patch assay, we found the increment of hair follicle induction in ORSCM treated mouse dermal cells. Also, we found increment of hair follicle induction in ORSCM treated red vibrissa dermal papilla cells using chamber assay. So next, we supplemented ORSCM to human dermal papilla cells. As a positive control, we used mouse epidermal cells and mouse dermal cells. And, as expected, human dermal papilla cells in conventional culture, 10% FBS supplemented DMEM with mouse epidermal cells did not induce any hair follicles. To our disappointment, we have not, the trichogenicity of human dermal papilla cells has not been increased by ORSCM treatment. Recently, vitamin D3 analog calcitriol treated red dermal papilla cells induced hair follicles. So we tested this to human dermal papilla cells. Human dermal papilla cells responded to calcitriol, so Wnt10B and alkaline phosphatase and SOX2 expression had increased by calcitriol. However, calcitriol treatment to human dermal papilla cells did not recover or maintain trichogenicity of human dermal papilla cells. Also, BMP6 treatment cultured human dermal papilla cells responded to recombinant human BMPs. However, another disappointment, human dermal papilla cells, treatment of BMPs up to 400 nanogram per ml did not maintain or recover trichogenicity of human dermal papilla cells. Recently, Oyama et al. Reported that human DP signature genes, and they found that Wnt, BMP, and FGF signaling pathways are upregulated in intact dermal papilla. And they developed DP activating culture medium containing bio GSK3 beta inhibitor to activate Wnt pathway and BMP2 and BFGF. So they found that impaired DP properties in conventionally cultured human DP cells are restored by this medium. So they found hair follicle-like structure. So we tested whether DPAC medium can maintain trichogenicity of cultured human dermal papilla cells. So you see, so this is passage 2 human dermal papilla cells with. So we, from starting culture, we maintain these human dermal papilla cells in this medium, and there we found some trichogenicity maintenance. And CMDII labeling of DP cells before implantation showed that human dermal papilla cells are in the dermal papilla of induced hair follicle. Recent studies also showed the spheroid culture to maintain trichogenic dermal papilla cells. So cells, and especially for human scalp dermal papilla cells, we made spheres with low cell binding plate and hydrocell plate, and we found that in contrast to the flat dermal papilla cells, passage 10, spheres, human DP spheres with the mouse epidermal cells induced hair follicles, and CMDII labeling showed that implanted DP cells are in the DP and dermal sheath layer. Also, very recently, Higgins reported that 3D cultured human dermal papilla cells placed between the epidermis and dermis of neonatal foreskin induced hair follicles by sandwich assay. Next, we asked whether restoration of trichogenicity by sphere formation is unique to dermal papilla cells or can be applied to adult dermal fibroblasts. This will be presented this morning by Dr. Kwan. But briefly speaking, in contrast to dermal fibroblasts, 3DDF cells did not induce any follicles, as examined by patch assay and chamber assay. We also asked whether restoration of trichogenicity by sphere formation can be applied to other adult stem cells, such as adipose-derived stem cells. This is poster presented by Chang Seo. To our disappointment, ADSC spheres hardly induced hair follicles. as expected, trichogenicity indicators are restored by DP sphere formation. And we performed microarray analysis and found that a number of genes are unregulated in 3D trichogenic DP cells. Our data and data from Higgins showed that some of DP signature genes, including trichogenic genes, are restored by sphere formation. So we examined whether post-expression of putative trichogen gene, such as like-1, it's a transcription factor gene regulated by beta-catenin. And so we cultured mouse neonatal dermal cells were transduced with control lentivirus or overwhelming expressing lentivirus, and we performed patch assay. So we found enhanced hair follicle neogenesis by post-expression of overwhelming. So because overwhelming is a transcription factor, we studied the downstream target genes. And this was really this is presented by Dr. Park. We found two genes which are regulated by overwhelming transcription factor, and we found that ablation of overwhelming target genes by siRNA method, we found that ablation of these target genes impairs hair follicle induction. Okay, this is a summary. In vivo animal models are available to evaluate trichogenicity of cultured cells, and trichogenicity of DP cells can be maintained or restored by supplementation of optimal vectors to culture condition and sphere formation. And restoration of trichogenicity by sphere formation is not applicable to adult dermal fibroblasts and adipose-derived stem cells. And post-expression of trichogenic genes is a promising option for restoring trichogenicity. So I thank my former students and colleagues at the KNU School of Medicine and Professor Kim's in Hair Transplantation Center and Dr. Hwang in Kuwait for lentivirus construction, and Professor Lee in Chai University for adipose-derived stem cell supplying. And this study was supported by Brain Korea 21 and BK 21 Plus programs, and also funded by Korean Ministry of Education. Thank you for listening.
Thank you very much. Unfortunately, you spent all of your time. Sorry, so you can't provide.
Sorry, I'll be happy to discuss. Afterwards. Yeah.